Alrighty. So, hello everyone, welcome. I know it's been a long day, so I want to introduce you to my research. So for this summer, I've been identifying the role of host factors in the human papillomavirus. As Brenda said, my name is David Rodriguez. I'm a rising senior here at the University of California, Santa Barbara. My PI is Jan Carette. My mentor is Allie Dupzyk, and I'm part of the SSRP AMGEN Scholars Program. So many of you may be wondering, what is HPV? What is the human papillomavirus? Well, it is a small, non-enveloped, double-stranded DNA virus, and it's actually one of the most common sexually transmitted diseases. And it spreads through skin-to-skin contact, and specifically through breakages in the skin. And here, the infection will begin in a breakage where the virus enters down into the basal layer, or the bottom layer of the skin. And there's many types of human papillomaviruses. There are actually 450 that are currently known, and not all of them cause health problems. Most of them are actually asymptomatic, and they don't cause symptoms at all, and actually resolve themselves on their own. However, some cause genital warts, and then there's only a handful that are high-risk human papillomaviruses that are oncogenic, which leads me to say that HPV causes cervical and oropharyngeal cancer. And actually, HPV accounts for 5% of cancers per year worldwide. And almost all cervical cancer is caused by HPV. 9 out of 10 times, if a patient goes into the hospital for cervical cancer, it is due to this virus. And conversely, on the rise, there's been around 60 to 70% of oropharyngeal cancers of the mouth and throat that have been caused in men by this virus. And I want everyone to take a quick look here of what the virus looks like. We have two capsid proteins, which we call L1 and L2, and then there's also a genomic DNA in the middle. And for my research and for my experiments, I'll be using a pseudovirus, and this virus will be used as a tool to study HPV entry. HPV itself, or this pseudovirus itself, will not cause a true infection, but will still go through cell entry and everything we need to know to study it. Here's a schematic of my virus. Here we have our L1 capsid protein, and we have our L2 capsid protein. Additionally, these two capsid proteins surround a reporter plasmid. And this reporter plasmid will contain our GFP, or green fluorescent protein. And this is really important because this will tell us if there's successful entry into the nuclear envelope. GFP will only be expressed in the nucleus. Here we have microscopy of the L1. We can see it's on the outside, and we have L2. And I'm pointing this out a lot because L1 and L2 will go through conformational changes through proteases that are important for cellular entry. However, in current research, HPV entry is complicated, highly debated, and much is actually unknown. But from what we do know about HPV entry, it begins here, at attachment receptors called HSPGs, where binding first occurs. And it's here we see those proteases cause conformational changes. And then here we have an unknown secondary receptor, which will allow for endocytosis, or entry into the cell. And next, what we see is entry into the cell, and then an enzyme named gamma secretase. Gamma secretase is really cool in the sense that it will help L2 be brought to the cytosol, which we see here. And we kind of see the virus almost looks like a lollipop with gamma L2 on the outside. Additionally, we have retromer, another enzyme that attaches to the microtubule that traffics the virus to the trans-Golgi network. And it's here the virus will stay until mitosis, or cellular division, where the virus will then enter when the nuclear envelope breaks down. So, with our pseudovirus, we'll begin using a CRISPR-Cas9 screen to identify important host factors for cellular entry. And this will first be done by using a cell library with genome-wide CRISPR knockouts. What this means is we have a lay of the land of cells, and we'll use CRISPR to knock down different proteins and host factors in these cells. We'll add our virus, our HPV16, and that will infect these cells. And this will help detect genes that are important for infection, because some will be infected and some will not, because we knocked down those genes. And what we found is that there were many host factors, many. But we'll only be focusing on two for my summer research. And here we'll be focusing on TMEM253 and SEMA6D. And these two host factors are genes involved in the cellular membrane. TMEM253, or transmembrane protein 253, has been predicted to be a critical component of the plasma membrane. Additionally, SEMA6D, or SEMA4 and 6D, is part of a large family of proteins. and they're both include secreted and membrane-associated proteins. And lastly, we'll be using these host factors to create a CRISPR-Cas9 pulled knockout. So to begin our pulled knockouts, we'll be creating a recombinant plasmid. We'll be generating a lentivirus. So step one of this will be starting with a plasmid. And here we'll be using molecular scissor using restriction enzymes, which will create a place for our oligos. And oligos will contain our host factors for our knockouts. And then through ligation and our plasmid digest, we'll have our plasmid with its annealed oligos, and we'll be able to do a transformation using heat shock into our bacteria. Here, our bacteria will then create our oligos, our plasmids on a larger scale, in which we could use a purification process to create our plasmid library with our knockouts. Once we have our plasmids, we can use transfection into 293 FT cells. These cells are really important because they'll be generating our lentivirus. These lentivirus will be a genetic vector for our plasmids. Next, with our lentivirus, we will transduce our plasmids for pull knockouts into HeLa S3 cells. Here we have our lentivirus created by our cells, thank you cells, and we'll be using transduction into HeLa S3 cells. Here we'll be able to select our cells and the ones that are truly infected with our lentivirus using puromycin. Puromycin is just an antibiotic. Next, we'll use our HPV and add it to our HeLa S3 cells. Remember, we have that reported plasmid with GFP. So if there's successful nuclear arrival, we'll see fluorescence, and we'll be able to measure this using flow cytometry. Next, with our GFP, we'll be able to measure this in flow cytometry and observe infection. Here we have a positive control. It says wild type, but here we knock out genes such as gamma secretase or retromer, which were important for cellular arrival. And here we get a nice, perfect positive control of 0%. Conversely, we have a negative control where you have normal HeLa cells and HPV16, and we get a baseline of around 85% cells. And then just for these graphs, forward light scattering is size, and GFP is brightness of fluorescence. So when we looked at SEMA60 and TMM253, and we knocked them out, these host factors that we're observing, they actually had very similar infections to our negative control. So which means we had around 91% of our cells fluorescing and around 94% of our cells fluorescing. And this data we generated onto a bar graph, and it's probably likely to say they're not important host factors for HPV entry. Here we have our controls, our positive controls, and our gene knockouts, and our percent of infection. And we see that our relative base infection of 100% was very similar to our knockouts. So they were being infected. But that's all right. If we did find important host factors later on, we could do rescue experiments. We could use and do gene add back. So in the immediate future, if we did have a host factor that was important, we could do a Western blot. It showed that this gene is actually disrupted. Or we could do a gene add back, where we knock out a gene, watch this infection go down, and restore infection again. But what would this look like? So it would kind of look something similar to this, where we have a host factor of interest, and then try to prove it's important through gene add back. So here, similar to what we did, we'd have a wild type gene with a 100% infection, and then, we have a successful host factor, we'd have decreased infection. And then additionally, we'd be able to add back this gene, and we'd have infection go to either baseline or close to it. And I just also want to make sure to point out this isn't actual data, this is something hypothetical of what it looked like. And then in maybe the distant future, when we were continue this study, we'd be able to do interaction studies. How would one host factor look with another? Protein-protein interaction. How about the location of proteins? We said that there was unknown secondary receptors, and this would be really important to look at. And lastly, our research seeks to help fill a gap in the limited understanding of HPV cellular entry. And our overall goal this summer was just to identify host factors essential for HPV infection. And this was done so we could reveal novel targets for antiviral therapy, but also achieve our goal and objective of combating HPV-induced diseases like cervical cancer and oropharyngeal cancers. And lastly with that, I'd like to give my acknowledgements. I want to thank my mentor, Allie. She's been so patient, so amazing this summer. I'd like to thank my PI, Jan Carette. Without him, I wouldn't be able to do this research. And lastly, I want to thank all the lab members and the SSRP program leaders in the cohort. Without all of you guys, I wouldn't have this supportive network and be able to do what I am doing today. And with that, thank all of you. I'm now open to questions.
Great presentation. Very, very kind to people that are not from this world. But yeah, I was trying to get a sense of like just how many of these human factors are there, like a scale of like things you have to check out before you're like going into the right one.
Oh yeah. I wish I knew like a number to tell you around the top of my head, but when my mentor was showing me all the host factors we found, knockouts, it's like a list. It's like a tier list almost. You have one host factor that has like a bunch of knockouts, like no infection, and then kind of you have ones that go over. And then we have like a baseline of like ones that are important host factors and ones that are just kind of throwaways. But a lot of these host factors, if you knock them out, the cell dies anyways. So you know, like one might be important for the nucleus, or you know, important for protein synthesis. So after ignoring those, you get maybe like random guess. But maybe 20 or so host factors that we could look at. This summer we actually looked around 6 with between me, Pearl, and my mentor.
Hey David, great job, proud of you. So I have a quick question. So I work in cancer bio, but you also said that some human papillomaviruses are oncogenic and some are not. Why is that?
Ooh, good question, good question. So like I said, there's 450 known human papillomaviruses, and actually only a handful, I think there's actually 9 that are oncogenic, HPV-16 being one of them. And HPV viruses, papillomaviruses, they're very similar in structure. They have L1, they have L2, they're proteins and stuff like that. But what's different between them is their viral genomic DNA. So there's these factors in their viral DNA like E6, E7 that are important that make that specific human papillomavirus oncogenic. And that's what made one cancerous or oncogenic.
Alright, hi David, great talk. I noticed that in your chart of results for like the results that you found, there were in the bar graph it was like over 100%. How was that like calculated, or how did you find that?
Let me see if I can go back here for you. This one, yeah?
Yeah.
So here, this wild type, that negative control we looked at with regular, it was around 80%, 86% of cells fluorescing. So that was our baseline, because this is our control. So that's set to 100. And then these, when we knocked out Team M253 and Team 60, we actually saw higher, around 90% infection. And this was kind of an odd result, because if you knocked out something like that's important for the pathway, why would you see increased infection? But what we saw is that when we knocked down these genes, increased infection occurred. So that's why you have something over 100%.
Do you have any like theories as to why that occurred?
Maybe a theory to this would be the sense that it's a cellular membrane protein. HPV spreads through division. Maybe there could be increased cellular mitosis that's been caused by the knockout. But not sure to say. I guess that's what our research is here for. And it's also important to say Team M253 is a recently found protein. It doesn't even have a name yet. That's why it's transmembrane protein 253. It's not 252, right? So we're still very
Yeah, over here. Yeah. great talk. Really cool. Yeah, and actually I'm really interested, sort of piggybacking off the point that Martine made about Team M253. Seeming as like it's unknown exactly sort of what its function is. I guess I'm curious to see like if you had extra time, would you investigate these more? Would you try and find out their mechanism, or would you try and look at other proteins to see if they play better roles in that as well? Infection.
Great question, and that's actually one of the first things I talked about with my mentor when we found our results, is like, is this something we should pursue more, is this not? I think it's something you shouldn't really disregard, because when you get results like this, especially after we did a screen that, these knockouts worked and, decreased infection, that's why we found them important. So, sometimes when we get results like this, it's actually even more important, it's intriguing, so maybe you should look into it, because why are you seeing such a breakdown like this? And the next steps I would look at, especially with these, if I was to pursue it more and I had more time in this summer, it would definitely be protein-protein interaction. If you're just knocking out one, maybe it's not doing it for the cell or the virus, but if you knock out 2 together, that's where it gets interesting.
Thank you.
Mm-hmm.
So I have a quick question about, I don't know if I missed this, but with respect to the screen and kind of like you testing two specific proteins, like sometimes when you're doing CRISPR screen or something, or a screen for anything, your screen conditions might not be screening specifically for what you're looking for, and then when you follow up on it, maybe the conditions have changed a little bit, and so maybe you don't get the same phenotype or result when you follow up. So do you think that your follow-up analysis was the exact same conditions, or do you think there could be some variables that changed which didn't allow you to see the phenotype that you saw on the screen?
Great question. So I didn't personally see the genetic screen that was first done in that one step I spoke about. But between at least Team M2V3 and Team M60, these are the same results. Same HeLa cells, same plasmid, plasmid recombination that we made. So our variables, at least when doing the HPV infection, was the same. However, with the genetic-wide CRISPR-Cas9 screen, it's a different procedure objectively. So I think that may be one reason why we may not just see that result, and I would have to look more into it. But great observation.
Great. If there's no more questions, let's give David another round of applause. And that concludes our SSRP symposium.